All right, so today I'm going to talk to you about proteins, and much of this should be review because I've talked a little bit about protein structure when I was describing some of the different representation styles for different kinds of molecules. There's unique things like ribbon models and backbone models that are unique to proteins. So we discussed some of that already. And then in the latter part of today's class, I'm going to introduce Chimera, which is another one of these molecular visualization tools, and have you do a couple simple exercises in Chimera. So as far as protein structure is concerned, it should all be review at this point that proteins are built from smaller building blocks called amino acids. Once again, here's the basic structure of an amino acid. There's a carbon in the very center, referred to as the alpha carbon. There's an amino group or amine group, NH2, on one side, carboxylic acid group, COOH, on the other side. Typically, there's another hydrogen attached to that alpha carbon, and then there's a side chain represented by the letter R. And it's the side chain that differs from one amino acid to another that makes each amino acid unique. Here's a list of the 20 amino acids that are encoded in the DNA. And what I mean by that is that there are actual codons, a sequence of 3 bases in the DNA and hence in the messenger RNA that code for specific amino acids. There's an anticodon on a transfer RNA molecule that delivers a particular amino acid to the ribosome. There are actually other amino acids besides these 20. There are other amino acids that are found in biological molecules, but those other amino acids are things that are modified after the protein has been constructed at the ribosome. So these are the 20 that are actually coded for in the genetic code that are strung together at the ribosome, and then some of them may be modified further at a later time. These 20 amino acids all have a 3-letter code, which I actually know all of these 3-letter codes, and I think it's a good idea for everybody in our profession to at least be familiar with a lot of these 3-letter codes, be able to recognize them. There's also a 1-letter code for each amino acid, and honestly, I've never bothered to try to memorize those. Sometimes these are used if you want to show the amino acid sequence of a protein, rather than using a whole bunch of 3-letter codes strung together, they'll just put a whole sequence of single letters together to represent the sequence of amino acids in a protein. There's a couple here that you don't see used very often. There's this thing ASX, or represented by the single letter B, which can refer to either asparagine or aspartic acid. There are actually separate codes for those individually. So ASP is aspartic acid, and ASN, you see over here on the right, is asparagine. But there's this generic one, which means that that particular position could be occupied by either asparagine or aspartic acid. Same thing with glutamine and glutamic acid. There's a generic one for those as well. But I don't see those very often when I look at amino acid sequences. As I said, the thing that distinguishes the amino acids from one another is the side chain. So if you look at all of these amino acids, all but one of them have that same basic structure that I was talking about before. So for example, if we look at glycine up here in the upper left, you see the alpha carbon with the NH2 amino group attached to it on one side, the extra hydrogen, and then the COOH, the carboxylic acid group, on the other side. Now, in the case of glycine, the R group, the side chain, is literally just another hydrogen atom. So it's the simplest of all of the amino acids because its side chain is just an H. Then alanine, the side chain is a methyl group, CH3, and you see that some of these get progressively more complicated with branched alkyl chains or branched carbon chains off to the sides. Some of the larger amino acids, some of the more complex ones, actually have benzene rings in their side chain. So phenylalanine, it's basically alanine with that methyl group, but then with a phenyl group, the benzene ring attached to the methyl group of the alanine. Tryptophan has this double ring structure in its side chain. One of the more unique amino acids is proline, that does not have that basic alpha carbon structure with the amino group on one side and acid group on the other. The alpha carbon in the case of proline is actually part of this five-sided ring. And it doesn't have a full amino group. It doesn't have NH2. It just has HN in place of an amino group. It does have the carboxylic acid group up here. But proline is one that has a radically different structure from any of the other amino acids. And that's actually important in understanding protein structure, because the presence of a proline amino acid in an amino acid chain puts a definite kink, a bend in the amino acid chain, because it doesn't follow that standard structure. All of these amino acids that I just mentioned are all hydrophobic amino acids, meaning the side chain is non-polar. There's no electrical charge. It doesn't have a plus or minus charge. It doesn't have even any polarity to it. There's nothing, what we would call a dipole. There's no polarity in the side chain. The fact that these particular amino acids are hydrophobic is important because we find hydrophobic acids tend to reside closer to the center of a protein structure because they are repelled by the water that surrounds the protein. Also, you find a lot of these amino acids with nonpolar side chains in a portion of a protein that might be embedded in the cell membrane of a cell, because the cell membrane has a hydrophobic part. All those lipid tails are hydrophobic. So you would find a lot of hydrophobic amino acids embedded within the membrane. So if a protein is actually embedded in the cell membrane, it would have a transmembrane portion that might have a lot of these hydrophobic side chains. We talked a little bit about a color scheme once before for coloring amino acids. All of these that have nonpolar side chains are colored with dull colors. So sort of muted grays and beige colors and things like that. Then there's a group of amino acids that are very, very polar. They have not only polar side chains, but they actually have a full charge to them. There are two that are acidic, glutamic acid and aspartic acid. There would be an OH down here, but the H has dissociated, leaving an oxygen with a full negative charge. So these are considered polar, and they actually have a full negative charge to them. There are also a couple of amino acids that have an additional NH2 group, an additional amino group, which can attract another hydrogen to become positively charged. So lysine and arginine actually will have a full positive charge in their side chain. So these are also, very highly polar. In fact, having a full positive charge. Anything that has a charge like this is going to be attracted to water. So it's going to be hydrophilic. So these amino acids would tend to be closer to the outside of a protein structure because they'd be attracted to the water on the outside. And you would not typically find these amino acids in a portion of a protein that was actually embedded in the cell membrane. These more hydrophilic amino acids would be more likely, actually out facing towards the cytoplasm, or excuse me, facing inward towards the cytoplasm or facing outward towards the extracellular fluid. Anything that has a polarity to it is colored with brighter colors. And in the case of these that have a full charge on them, the two acids, that negative charge is coming from an oxygen. So since oxygen is normally colored red in the CPK coloring scheme, these two amino acids are colored bright red because of that negatively charged oxygen. And then conversely, the two positively charged amino acids, the positive charge is coming from an amine group, which has nitrogen in it. And since nitrogen is blue in the CPK color scheme, these two amino acids are colored bright blue. And then finally, there's a group of amino acids that have polar side chains, although they don't have a full charge to them. They have what's called a dipole, where there's a little bit of a difference in charge from one side to the other, but it's not a full positive or a full negative charge. And these are all, they're still hydrophilic, because that polarity or that dipole in the side chain is still attracted to water. These are colored with warm or cool colors. If the polarity in the side chain is coming from the presence of oxygen, like serine, threonine, and tyrosine, then they'd be colored with warm colors, so oranges and that sort of thing. In the case of some of these others, that'd be a different kind of a dipole that's being produced primarily by the presence of these nitrogens in the side chain. These would be colored with cool colors, again, because nitrogen is normally blue in the CPK color scheme. So these would be like turquoise and greens and other cool colors. And then the last group of amino acids are the two that contain sulfur in their side chains. Cysteine and methionine have sulfur in their side chains. And the presence of the sulfur in the side chain is important also for understanding three-dimensional protein structure, because one of these sulfur-containing amino acids can form a bond with another sulfur-containing amino acid far away in the amino acid chain to actually bring two parts of the amino acid chain together to form what's called a disulfide bond or a disulfide bridge. And that can have a dramatic influence on both the tertiary structure and even on the quaternary structure, uniting separate amino acid chains together. These amino acids are always colored yellow in the amino acid coloring scheme because sulfur is yellow in the CPK scheme. So you remember that proteins are formed when these amino acids join together. The OH group from the carboxylic acid will break off and form a bond directly to the nitrogen of the amino group on a neighboring amino acid. That one of the hydrogens from the amino group breaks off. So you've got an OH from the carboxylic acid, a hydrogen from the amino group. They join together to form HOH, or better known as H2O, or water. So you expel a water molecule in the process of bringing these two amino acids together. That bond that's then formed between the carbon of the carboxylic acid and the nitrogen of the amino group, that's known as a dipeptide bond, or excuse me, actually, it's known as a peptide bond. And at this point, the molecule would be called a dipeptide because it's composed of two amino acids. And then this continues to create a long, long chain of amino acids where you're continuously adding new amino acids to the carboxylic acid end of the amino acid chain to ultimately form what's called a polypeptide, where there are multiple amino acids all joined together. Once the amino acids have been joined together as part of this chain, we refer to the residual amino acid, or essentially what's left of the amino acid, as a residue. So each amino acid, once it's part of this chain, it's lost parts of it. It's lost a hydroxyl group, it's lost a hydrogen from one side. So it's not a complete amino acid. That's why we use the word residue to refer to it in the chain. The definition that I've heard of a protein is that it's any polypeptide with greater than 50 residues, but that doesn't seem to always hold true, because I know of at least one protein that has fewer than 50 residues. So a protein is really just any long polypeptide chain. There's no real strict definition as to how many residues you have to have before you call it a protein. The polypeptide chain has a polarity to it. It has a beginning end, which would be the first amino acid that was laid down at the ribosome, and when the process of translation started at the ribosome. This is known as the N-terminus, because the beginning of the very first amino acid starts with the amino group, which has a nitrogen in it, so hence the name N-terminus. And then the very far end of the amino acid chain, the very last amino acid that was added at the ribosome, this is known as the C-terminus, because it's going to end with the carboxylic acid of the final amino acid in the chain, which has carbon in it, so hence C-terminus. So every protein or every amino acid chain has a beginning, which is the N-terminus, and an end, which is the C-terminus. So we've also talked a little bit about the different elements of protein structure. The primary structure of a protein is simply the linear sequence of amino acids the linear sequence of residues that was laid down at the ribosome. Immediately after that amino acid chain starts to form, it begins to fold to form loops and sheets and spirals called alpha helices. These are all elements of what are called secondary structure, where that amino acid chain begins to fold itself into these spirals and sheets and so forth. Something we didn't talk about before is that there's a level that's kind of between secondary and tertiary. There's something known as a supersecondary level. These are sometimes referred to as protein motifs. These are typically collections of secondary structures, but it doesn't represent the full three-dimensional shape of the tertiary structure of the completely folded protein. So I'll show you some examples of supersecondary motifs in a couple minutes. So then we jump from supersecondary structure to tertiary, where all of these alpha helices and beta sheets and loops and everything are now folded into one compact blob. And there are lots of proteins that this is all they have is primary, secondary, and tertiary structure. And you end up with a fully functional protein. But in other cases, you actually have to take multiple amino acid chains and put them together to form an even larger, more complex structure. And that's what we would call the quaternary structure, where you have a larger protein that consists of several amino acid chains joined together. So primary structure, as I said, is just the linear chain of amino acids. So here we just see some gray bars, which represent what we call the backbone of the protein. That's just, where you've got the alpha carbon, the carboxylic acid carbon, the amino nitrogen, and then that repeats itself over again with another alpha carbon, another carboxylic acid carbon bonded to a nitrogen, and so on to create this complete linear chain of the amino acid. And this is showing, of course, that chain is made up of lots of different amino acids, each of which has its own unique side chain, which is just shown sticking out to the side of the chain itself. And we've talked about some of these secondary structures before, how sometimes the amino acid chain will roll itself into a spiral known as an alpha helix, and that alpha helix is stabilized by the presence of hydrogen bonds between one of the oxygens, that carboxylic acid oxygen, and a nitrogen on a neighboring amino acid. There are 3.4 amino acids for one complete turn. So almost 3.5 amino acids makes one complete turn of the alpha helix. We've got beta sheets. Now, the type of beta sheet that I've talked about before in this class is what's known as an anti-parallel beta sheet, where the arrow represents the direction of the amino acid chain with the N terminus at the beginning and the C terminus at the end. And the amino acid chain travels in that direction from N towards C, then makes a sharp hairpin turn, and then goes back the other way, then makes another sharp hairpin turn, goes back the other way, and so forth. That can continue on for quite a ways. And then these chains are then held together by hydrogen bonds represented by the little dotted lines. And then this creates an entire sort of flat sheet, hence the name beta sheet. It's called anti-parallel because the neighboring strands are pointing in opposite directions. So one strand is going from N terminus to C terminus. The next one, that direction is reversed. So, we've got N down here and then C up at the top. There is also something called a parallel beta sheet, where an amino acid will go from N terminus to C terminus, and then there'll be a great big long loop that loops all the way back around, and then another portion of the amino acid chain will also go from N terminus to C terminus, and then another great big long loop, and then another segment of the beta sheet. So this also forms a broad sheet, and it's all stabilized by the presence of those hydrogen bonds shown by the little dotted lines. The difference here is that the neighboring strands are actually parallel to one another. The N terminus to C terminus direction is oriented in the same way. There's also a beta turn. which is that tight hairpin turn that you find in an anti-parallel beta sheet. Then there are some non-repetitive structures that just sort of random loops and bulges and random strands of the chain that are all components of this secondary structure. The secondary structures are then packaged into this other level of structure that, I just introduced to you today. We haven't talked about these before. These are known as supersecondary structures, or sometimes called motifs. There's one that's called a beta alpha beta unit, sometimes represented by this big letter N. What this is showing is that you've got a beta strand, part of a beta sheet. It comes up, there's a tight turn, then an alpha helix, then another tight turn, and then you go back to another part of a beta sheet. Apparently this beta alpha beta unit is a fairly common motif that you find in a lot of proteins. There's also one called a Greek key, which is basically a beta sheet, but if you look at how it's formed, you've got a strand going from N terminus to C terminus here. Then there's a big long loop that connects over to a beta sheet that's some distance away, and then a tighter turn that loops up next to a beta strand that's a little closer, and then zigzags back and forth. I often think that this looks like a paper clip, the way that piece of wire is folded to create a loop inside of a loop in a paperclip structure. This is known as a Greek key because if you look at the purple representation of that pattern, this is a common geometric pattern that's found in a lot of Greek art, particularly a lot of Greek pottery, ancient Greek pottery. And then finally, another example of a supersecondary structure is a beta barrel, which is basically just a beta sheet, but the beta sheet is wrapped around to form a complete cylinder. That flat beta sheet is wrapped around in a cylindrical fashion to form basically a large cylindrical opening. So we've also talked about tertiary structures, which is the next level of structure, where all of those secondary and supersecondary structures are now folded to form a compact blob. This was John Kendrew's low resolution model of myoglobin as he was starting to solve the 3D structure of the myoglobin protein. And each of these white blobs, it turns out, represents an alpha helix. And you can see there's a whole bunch of alpha helices all folded up to create this compact blob. And this would produce a fully functioning molecule of myoglobin. But then other proteins require multiple amino acid chains to come together. We've seen these pictures before. This is Max Peritz's model of hemoglobin, published in 1960. And it turns out that a single functioning molecule of hemoglobin requires 4 amino acid chains, 2 alpha chains, a beta chain, and a delta chain, if I remember correctly. So 4 slightly different amino acid chains have to come together to form one functioning molecule of hemoglobin. So we've also talked before about different ways of representing proteins, so this will be a quick review. You can represent a protein the way you would represent any other molecule. You could use a stick model where you see every bond. You could use a ball and stick model where you see, every single atom as well as the bonds between them. So this was the high resolution model of myoglobin that John Kendrew produced back in 1958, and a painting of that model by Irving Geis done shortly after that. You could represent proteins as space-filling or CPK or sphere models, where you see every atom represented by its van der Waals radius. You can have backbone models of proteins. Remember, this is review from just a few minutes ago, that the amino acids are all strung together in a long chain. And if you look only at the bonds between the nitrogen, the amino group, the alpha carbon, the carboxylic acid carbon, and then that peptide bond where the carboxylic acid bonds to the amino group, and then that repeats again, CCN, 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 CCN. When you create a backbone model of a protein, what you're doing is you're showing only the backbone, only that repeating CCN, 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 which shows you the folding of the amino acid chain, and you're omitting all of these side chains. So the side chains are being completely ignored when you create a backbone model. So here's a backbone representation of a protein called lysozyme, which you've seen this before. And each of these little straight segments represents a single amino acid residue. So it represents a nitrogen, the amino nitrogen, the alpha carbon, and then the carboxylic acid carbon. And so you just have that repetition of NCN or NCC, NCC, NCC throughout the molecule. Another type of representation that's used typically just for proteins would be a ribbon representation that emphasizes the secondary structures that are present within the protein. So alpha helices are represented by these corkscrew spirals, beta sheets represented by these arrows. And remember that the arrowhead is pointing in the direction from the N terminus towards the C terminus of the molecule. So just a couple different representations of ribbon models. Here's another ribbon model, this time of lysozyme, and a ribbon model of a potassium channel protein that allows potassium ions to pass through the cell membrane. Another type of representation for protein, it's not limited to protein. Theoretically, you could represent any molecule using a surface representation, but it's particularly common with proteins because you want to understand the three-dimensional structure of the protein in order to understand its function. Looking for binding pockets, binding sites on the surface of the protein molecule. So we create these solvent accessible surfaces digitally. They're done in a computer by taking an imaginary digital sphere of a certain radius and then kind of rolling that around on the surface to see where that probe sphere touches the surface of the molecule, as well as little tight areas where the probe sphere can't get into there. And so here you see a comparison of a CPK representation with a solvent accessible surface representation of lysozyme. Here's another solvent accessible surface representation of that potassium channel that I showed you earlier. If you could rotate this and look at it from the top, you'd see that there's actually a little pore that runs right through the center of the molecule, which is the little channel that the potassium actually passes through to get either into or out of the cell. I've mentioned this guy before, David Goodsell. I showed you one of his books way back on the very first day. He has his own very distinctive style of painting molecules, but it's all basically a type of surface representation. You can kind of make out the individual atoms, but he doesn't draw an outline all the way around each individual atom. So by not drawing the outlines all the way around, it reduces the clutter a little bit. So you still get a sense that these proteins are made out of individual atoms, but you're also getting a sense for kind of the overall three-dimensional surface structure of each protein. Another illustrator who has a very similar style is Graham Johnson. I showed you a book way back on day one called Molecular Cell Biology by, I'm drawing a blank on the guy's name now, but Graham was the illustrator for that book, and he did all of this artwork in Cinema 4D, whereas Goodsell does all of his in watercolor. These are actual paintings. Graham works a lot in Cinema 4D. So we've also talked a little bit about some color schemes for depicting proteins. So if you're showing every individual atom, whether it's a stick or a ball and stick or a CPK model, you can color the individual atoms by element the same way that you could with any other molecule. You could use this amino acid color scheme that I talked about a little bit before. So things like aspartic acid and glutamic acid, these are charged. So these are highly polar. They actually have a full negative charge in their side chain, and that negative charge is coming from an oxygen. So these are colored bright red, since red is the color of oxygen in the CPK scheme. Lysine and arginine are amino acids that have a positive charge in their side chain that comes from nitrogen, so they're colored blue, because blue is the color of nitrogen in the CPK scheme. Some of the other amino acids are polar, but they don't have a full charge. Tyrosine, asparagine, glycine, leucine, valine, isoleucine. So these have cool colors. And then your dull colors, like especially this dark gray for alanine, proline, they call it flesh color. And then they just have a category down here for others. Some of the other nonpolar ones would be this tan color. So these would be things that are nonpolar. And then you've got the two amino acids that have sulfur in their side chains that are colored yellow. So you could actually have a space-filling model of a protein, but color the atoms by residue. So anything that's red would be atoms that are part of either aspartic acid or glutamic acid. So these would be all of the atoms that are part of a single glutamic acid residue, for example, or aspartic acid residue. There's kind of an orangey color here. I don't think that's meant to be yellow. I think that's actually supposed to be orange. I don't see any yellow amino acids in here, so I don't see any of the cysteine or methionine, the ones that contain sulfur. You definitely see some that are, kind of dark gray and light gray, which would be nonpolar. You can also color a ribbon model by residue. So the little blocks of color on this ribbon model represent different amino acids, and again, following that color scheme. There are some in here that I can't quite tell if they're supposed to be orange or yellow, so I don't know if those are supposed to be, one of the sulfur-containing amino acids or just something orange, one of the polar amino acids. You can also choose to color proteins by their secondary structure. So you can use colors to highlight elements of the secondary structure, such as using red in this case to color all of the alpha helices, and then turquoise to color the beta sheets, and then just gray for any kind of random loop or coil that's not part of an alpha helix or a beta sheet. The space-filling models in here represent other molecules that are embedded within the protein. They're not actually part of the protein itself. And finally, one other very common color scheme for proteins is to color by chain. This would be something that you would use if you have a protein that has a quaternary structure, where there are multiple amino acid chains that have to come together to form a single functioning molecule. So this is IgG, one of the immunoglobulins, with the typical Y shape that we think of when we think of antibodies. But that antibody structure actually consists of 4 separate chains. There are 2 larger chains called heavy chains, shown in orange and red. And then there are 2 smaller chains called light chains, shown in blue and turquoise. So coloring by chain allows you to easily distinguish those 4 chains in this molecule. So something we haven't really talked about before is the classification of proteins. There are different types of proteins that perform different functions in the body. They have different structures, and those different structures reflect their different functions within the body. There are 2 broad categories of proteins. There are the fibrous proteins and then the globular or compact proteins. Fibrous proteins, the name kind of implies this, they are fibrous. They are long fibers or long chains. Oftentimes, these fibrous proteins consist of nothing but just long alpha helices, just long chains of alpha helices. They're not folded up into a compact blob of any kind. They're just long alpha helices that go on for long distances. These fibrous proteins are typically structural proteins. That they help create structure within the body. So collagen is a good example. This is a structural protein that makes up a lot of the connective tissues in your body. So fascia and tendons and muscles and cartilage contain lots of collagen, these long fibrous strands that give these tissues their strength. Keratin is the protein that makes hair, fingernails, deer antlers, rhinoceros horn. There's a lot of keratin present in the epidermis of your skin. There are actually cells called keratinocytes that produce keratin in the skin. It gives the skin a lot of its strength. So that's another example of a fibrous protein that has a structural function in the body. Then the globular or compact proteins are things that really do fold up into a compact three-dimensional blob. And there are lots of these. And this is by far not an exhaustive list. There are other kinds of compact proteins, other families, but these are just some examples of some of the compact proteins. Most of your membrane-bound proteins, the proteins that are embedded in a cell membrane, so receptors and ion channels and things like that, they are examples of globular or compact proteins. Most enzymes are globular and compact. Sometimes those enzymes are embedded in a membrane also, but other times enzymes are just sort of free floating in the cytoplasm. Transport proteins like hemoglobin that transport oxygen in the bloodstream, and myoglobin that stores oxygen in muscle tissue. And then there's lots of other categories of globular proteins. The antibodies that I showed you an example of, immunoglobulin G, IgG, just a minute ago. Those are proteins. There are histones, which are the proteins that the DNA wraps itself around. There's a group of, I'll show you a picture of it in a minute, but you've got a group of 8 proteins called histones, and the DNA double helix coils around these histone proteins to create something called a nucleosome, which in turn creates the structure that we call chromatin, which is how DNA is packaged in the nucleus. So I'll just show you some examples of these. Here's a picture of keratin. This is not an entire keratin molecule. This is just one small piece of it. And maybe a little hard to understand what you're looking at here. It's just a stick representation of the molecule, but you've got oxygen, which is part of your carboxylic acid. You've got the blue is nitrogen. That's your N, part of your amino group in one of the residues. You've got a alpha carbon. So there's a repetition of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. So you've got your linear amino acid chain, but maybe a little hard to tell, it's actually folded into a spiral. So keratin, the structure of keratin is basically just an alpha helix. Long strands of alpha helices that give your skin and your hair and your fingernails and stuff, give them their strength. Here are some examples of some compact proteins. These would both be examples of membrane-bound proteins. On the left is one example of an acetylcholine receptor. Turns out there are actually a couple of different acetylcholine receptors, but this is just one example of it. On the right is a potassium channel. There are also several different kinds of potassium channels. This is just one example. The little turquoise balls actually represent potassium ions that are waiting their turn to pass through the channel. The little flat layers of white balls actually represent the cell membrane itself. So you can actually see how these proteins are embedded within the cell membrane. And in maybe a week or so, I'm going to show you a really cool website called the OPM database. It stands for the Orientation of Proteins and Membranes. And what they've done is they've solved the structure of some proteins crystallographically to get their 3D structure. But when you download the molecular model files from the OPM database, they include those layers of white balls so that you can actually see exactly how the protein sits within the cell membrane, which if you're trying to illustrate a protein and you want to understand how that protein actually sits in the membrane, this OPM database is a lifesaver. I use it all the time. Here's a really boring looking enzyme. This is a classic example of a globular protein. I mean, it's just a blob. Doesn't even really have any distinctive shape to it, but it does have 2 heme groups embedded in it. So these are heme groups that are almost identical, probably exactly identical to the heme groups that you find in hemoglobin. So it's just a carbon and nitrogen ring. And that becomes then part of the functional group of this particular enzyme. So the heme groups kind of indicate that that's probably where the binding site is, where the important part of this enzyme is located. This particular enzyme is called cyclooxygenase. It's an important protein in regulating inflammation and pain. When there's damage to cells, the cells release a lipid called arachidonic acid. And then the cyclooxygenase enzyme converts arachidonic acid into a molecule known as a prostaglandin, which is an important mediator of inflammation and pain. So this enzyme is also the target for a lot of your non-steroidal anti-inflammatory drugs like ibuprofen and acetaminophen. Those drugs block the binding sites on cyclooxygenase and prevent the conversion of arachidonic acid into prostaglandin, so that helps to reduce inflammation and pain. I've already shown you this picture of IgG, just another example of a compact or globular protein. This one's a little hard to see what's going on here, but this is showing another example of a type of compact protein. These are the histones that I was mentioning earlier. So maybe you may be able to tell that what we're looking at here, the structure that's shown as a space-filling model is actually the DNA double helix. And I've actually drawn a little line on here so that you can tell that the DNA helix comes down and coils around and wraps around and comes back and wraps around again and comes back up around on the other side. So you've got the DNA double helix there that's wrapping the 1 3/4 turns of that double helix wrapped around a group of proteins in the center that are represented in this case as ribbon models. And there's 8 of them. This is known as the histone octamer. The proteins are called histones, and octamer just means there are 8 of them. So there's a green one, an orange one, a red one, turquoise one, purple one, a darker green one. I'm not sure you can see all of them in here, but so there's a group of 8 proteins, and the DNA double helix wraps around these 8 histone proteins to form a structure known as a nucleosome. The nucleosome, in turn, is then packaged with other nucleosomes to form the thing that we call chromatin, which is how DNA is actually stored in the nucleus. Important thing to think of, and sometimes we lose sight of this because, you know, we can easily go into a database like the Protein Data Bank or the OPM database, and we can retrieve these 3D models of proteins and create beautiful renderings of them. But keep in mind that if you're illustrating a picture of a protein, you're illustrating only one state of that protein, only one form of the protein shape. Proteins are not static. They don't typically just retain the same shape the whole time. Some do, but many of them are very fluid, and they actually change shape pretty dramatically in some cases. Here's a good example. This is the spike protein from the coronavirus. And there are actually 3 different forms of this. On the virus itself, it can have either the closed state or the down state, or it can have what's called the open state or the up state. And you can see that one part of the molecule in this up state is actually kind of tilted up. And the closed state is sort of the normal resting state for the spike protein, but when it's getting ready to bind to ACE2 on the surface of one of your cells, you know, when it's getting ready to bind to a different protein on one of your body's cells, it switches to this open state. And then I'm going to show you a YouTube video. That shows you how the protein changes even more dramatically after it actually binds. And I don't know if there's volume on this or not. I'll see. Okay, we got an ad for taco seasoning, so I guess we're stuck watching that. Okay, so here's our spike protein. That pink ball up there is the virus. And there's angiotensin converting enzyme type 2, or ACE2. And there's actually a molecule on the surface of your cells that interacts with the spike protein and cleaves part of it, cuts off part of it. And look at what's happening with these alpha helices on this protein. It becomes like a little strong man. It's literally pulling the virus down so it fuses with the surface of one of your, okay. I want to replay that one for you real quick. Yeah, I guess, I don't know, either there's no volume or I can't get it to come up on here. So there's your ACE2 receptor. It helps cleave part of the spike protein off, and then you get all these alpha helices that kind of unwind and create this cable essentially that anchors the virus to your cell, and then it drags the virus down to allow the membranes to fuse so that the virus can then dump its genetic material into your into one of your cells. And there's another quick video of that as well. So this is a really extreme example showing how the proteins change shape.
The surface of the virus is studied with spike proteins, colored team, membrane proteins, yellow, and envelope proteins, colored blue. Cross section shows the viral RNA gene orange, and the nucleocapsid protein hexagon. Individually, the effect of the SARS-CoV-2 giving airborne viral particles. On entering the airways, the virus particles into the respiratory vessels and treat them in lungs. SARS-CoV-2 spike protein is a membrane-bound protein on the surface of the cells in ACE2, shown here in front. Once bound to ACE2, the spike protein is unable to open, close, and freely, blocking it in place. Another cell surface protein, transmembrane protease series 2, shown here in orange, cuts the spike protein in a specific location. These segments of the spike protein fall away and expose previously hidden parts of the spike. The spike protein undergoes a series of dramatic conformation changes. During the first stage, the spike protein inserts itself into the cell. Once inserted, the spike protein folds back on itself, pulling the membranes of the cell and virus together so they can fuse. After fusion, the viral RNA is deposited into the host cell, where it hijacks the cell's machine to produce more virus particles.
Living with plaque psoriasis.
Okay. Don't need that. Okay, so that's my whole lecture about proteins. You know, pretty quick review, other than a few additional little things at the very end. We'll be talking a lot more about proteins over the remainder of this course, because they're very dramatic things to illustrate, and you know they're so important to regulating so many functions in the body. So why don't you all take a little break, come back about 3:05 thereabouts, so take like a 10-minute break, and then I will start introducing you to Chimera, another molecular visualization tool. Don't need all that volume on anymore. All right, so I'm going to show you the molecular visualization software called Chimera. And if you click on the applications folder, come up to our Perkins SciViz folder, you'll see there's actually two different versions of Chimera. There's regular Chimera, and then there's Chimera X. I'm just going to stick with regular Chimera for now. Chimera X has a little different interface, and I've been told that one advantage to Chimera X is that it will open larger molecule files that sometimes Chimera struggles with. But I'm much more familiar with the interface for regular Chimera, so I'm just going to stick with that for now. But if you want to play around with Chimera X, you're welcome to do that. If you want to download it for yourself, both of these are free. So, feel free to download them on your own computers. I'm pretty sure they run on all platforms, Mac, Windows, whatever. So I'm going to go ahead and open up Chimera. The full name of it is UCSF Chimera, just referring to the fact that it's produced by the University of California, San Francisco. That's why it's free. It's an academic thing, or at least I should say it's free to use for any kind of non-commercial academic purposes. If you're trying to use it for commercial purposes, they do actually prohibit that in their licensing agreement, and theoretically you're supposed to actually buy a subscription software from them if you're going to use it for something commercial. All right, so here's a basic interface. You can go to the file menu and say open, and open a file from there. I've actually got this computer set up so that it automatically opens every PDB or mol file in Chimera. If I select any file in here and go to the file menu and say get info or Command I, you can go down here where it says open with, and you can actually choose the default program to open a particular type of file. So I've already got this set up so that it automatically opens any of these PDB files in Chimera. So here we are with our sphingomyelin molecule again. I used this as an example last time. If you just click with the mouse and drag, it rotates it around in 3D. To zoom in and out, just use the Command key, hold down Command, or if you were working in Windows, you just use the Control key. Just click and drag. You can use the Option key, which I think is called Alt in Windows. Option to just translate, move the molecule around different places. There's an Actions menu up here, which is where you will control the appearance of your molecule. This is where you can change the different kinds of representations. It's a little different from PyMol. You remember in PyMol, there were those two buttons. There was an S button and an H button. S was to show different representations, and then H was to hide different representations. You could show, sticks and space filling and surface and all those things simultaneously. Then you had to hide each one individually. Chimera is a little different. There's just one set of menus up here for the different representations. So under Actions, Atoms, and Bonds, I could hide all the atoms and bonds, in which case we're not going to see anything. It's going to disappear, but it's still there. We just have to go back to Atoms and Bonds and Show to make them visible. And then as long as they're visible, we can change it to a stick representation, which it is now. Ball and stick. If I zoom in on it, you can actually see the little balls in a ball and stick representation. Or I can change that to a sphere representation, which is what they call a CPK or space filling model. They also have a wireframe representation, sometimes called a line representation, which I don't find particularly useful. Let me go back and stick with the sphere representation. A couple other things under here, under Atoms and Bonds. If I use the wire representation, let me go back to that. I can control the wire width. So I can actually give that little thin wire a little bit more thickness, but it's still not that thick. It's still not that noticeable. So I tend not to use that. Go back to sphere here. There's also something called rings. Remember we talked about the fact that benzene is a ring of 6 carbons that are sharing a bunch of double bonds. Sometimes the way that's represented is to have a little hexagon with a circle in the middle. That's what this aromatic disk and aromatic circle mean. Aromatic circle would add a little skinny circle to the inside of a benzene ring. I don't have a benzene ring open at the moment, so I can't really show you that. But some of these other representations are kind of meaningless. You know, ribbon representation doesn't mean anything with this kind of molecule. That'll only become more important when we open up a protein in a few minutes. I could put a surface on here, just have to say surface show. Not very interesting, kind of like in PyMol, you could add a surface even to a smaller molecule like this. Similar to PyMol, you can also change the transparency of that surface. So a higher number actually means more transparent. So now there's a transparent surface. Doesn't show up very well, but if I change the atoms and bonds to like a ball and stick representation, now you can see that transparent surface. Or I can then choose to just hide the surface. So you just toggle back and forth between any of these selections. You also use the actions menu to control colors. And if I just choose a color like red, it's going to literally color the entire molecule red. If I wanted the CPK color scheme, where carbon is dark gray, hydrogen is white, nitrogen is blue, oxygen is red, and so forth, I would go to the color menu here and come down here to color by element. And now you can see I've got gray carbons, white hydrogens, blue nitrogens, red oxygen, and a orange phosphorus. There's more you can also do with the color menu here, but if you select actions and color and you go all the way down to the very bottom of the color menu, there's this thing called all options. And what that's going to do is it's going to bring up a separate window for my color actions. So I can actually do the same thing in this menu. I could change it all to red. I could color by element. I could make it all magenta, whatever I want. So sometimes I'll just open this menu and use it for all of my color choices. Instead of going to the actions menu, I'll just use this this color actions menu and just keep it open all the time. One tricky thing to be aware of with this color actions menu is you have to pay very close attention to the right-hand side of this this little dialog box, or this little menu here. Notice it says coloring applies to, and then there's a series of little buttons that you can check. By default, it's set to this thing called all of the above, which means it's going to color the atoms and bonds, whatever color I choose. It's going to color any ribbons, if I had ribbons visible here, it's going to color those a certain color. Any surfaces that I had applied. I don't usually use labels, so I kind of ignore that. But let's say that I had put a surface on here. Right now, the surface actually reflects the color by element. So, over the oxygens, the surface is red. Over the carbons and hydrogens, it's gray, and so forth. But if I say all of the above, and I just change this to say cyan, it colors everything cyan. The surface is cyan, the atoms and bonds are cyan, everything's cyan. But I could change this and say coloring applies only to atoms and bonds, and then I could go back in here and color by element again. So now the atoms and bonds are colored according to the CPK coloring scheme, but it left the surface in that cyan color. Or if I click just on surface, I can change only the color of the surface while keeping the atoms and bonds the same. So you just have to be aware of what you're coloring over here. I can also click this button that says background, and then I can make the background whatever color I want. So this is the only way to change the color of the background in Chimera. is to pull up this color actions dialog box, select background in the right, and then pick whatever color you want. You know, I don't recommend that one. You know, white, some kind of gray, black. Pick from either of those. Normally, by default, this is just set to all of the above. Just be aware of that if you're trying to color something specific. All right, I'm going to close this small molecule of sphingomyelin, and I'm going to open a protein molecule just to show you some of the things that are unique to proteins. If you want to close a molecule, we want to say close session. You got to go to the file menu and choose close session. If I just try to open another molecule, it's going to open it and drop it right on top of this one, and they'll be overlapping. So I generally want to close the session of one molecule before I open another. So then let me go into my proteins folder, and I'm going to open up, let me try the potassium channel. So when you open a protein, by default, they almost always open as ribbon models. But we still have the ability to represent the model in any other way that we want. If I go up to the actions menu and I go to ribbons, I can choose to hide all the ribbons. So there's still a few little things in here. Those little sticks that you see are actually part of the protein. It's a side chain of some of the amino acids that actually serves as kind of a regulator for allowing these potassium ions to pass through. So that is actually part of the protein that's visible. But I hid the ribbons, but I could choose to show all of the atoms and bonds. So now I have all of the atoms and bonds visible in a kind of a stick model. And looking closely at this, I'm realizing I got no hydrogens. For some reason, Chimera likes to use this tan for carbon. If I pull up that color menu, I can make the atoms and bonds. I can color them by element. Once I choose color by element, then the carbons are actually colored dark gray the way they should be. So I don't know why Chimera likes that tan by default. But I'm noticing that there are no hydrogens at all. So I'm definitely not seeing every atom in this molecule. There's a tool that allows you to add hydrogens. Pymol had something similar. In Pymol, it was under the actions button over on the right. But here in Chimera, you have to go to the tools menu, structure editing, add H, brings up this little dialog box. There are ways of altering it, but I just leave the defaults. And if I click OK, it should add, okay, give me some kind of error, but it still did most of what I wanted. It added a whole ton of hydrogen atoms that were missing from the original representation. Let's see. Instead of sticks, I could show ball and stick. You know, so now it adds all the little balls in there. I could use a sphere representation. Now I'm getting a CPK model. So we're kind of getting a sense for the overall three-dimensional surface of this protein. You can still see the purple potassium ions that are sitting in the little pore, the little channel in the middle of the molecule. I could hide all of the atoms and bonds, which hides everything. And then I could add a surface. Just go to Actions, Surface, Show. And so I could show it as a solid surface. I could make it a mesh surface, which would allow you to see, you know, if I still had the ball and stick inside, you could see that inside. or I could make it a dot surface. Like that, that just gives a little hint of the surface. I'm just going to go ahead and leave it solid. I can change the transparency of that surface. You know, again, then if I had the, say if I turned the ribbon back on, I could show the ribbons inside. I could come back over to my color actions, and if I select ribbons, I could maybe change the color of the ribbons to something that's going to contrast a little better with the color of the surface. I can also color those ribbons by secondary structure. That is not something that's available here in the color actions palette, though. I kind of wish it was. It seems like a simple thing that you could change over here in color actions, but you actually have to go back to the tools menu. And there's a submenu called depiction, and right up at the top is a selection called color by secondary structure. That brings up yet another little dialog box here. You can decide what colors you want to use for alpha helices. Strand here means beta strand, so part of a beta sheet. So you can actually click on these little colors, and it brings up a color picker, and you can change the RGB values to change that orange to something else, or you could click on this purple for the strands and change that. And then the coils just represent sort of random loops and coils. So you can edit any of those colors if you want to. But once you've chosen the colors you want, you just click apply. Now in this particular model, I only have alpha helices and then just random loops and coils. There are no beta sheets, no beta strands in this, so there's nothing being colored in purple. If I hide that surface, you can now see the ribbon representation here. And if I think of it, I'll pull up a different protein in a few minutes that would have would actually have some beta sheets in it, so you could see the coloring of the beta strands. All right, I'm going to go back. I'm going to hide the ribbons again. Turn my surface back on. I'm going to go ahead and make that surface completely opaque, so a transparency of 0 means completely solid. And let me show you some tools for changing the rendering of the molecule. Back to the tools menu, there is a submenu called viewing controls. And there are several options here under viewing controls. There's something called side view, camera, effects, lighting, and shininess. I'm going to click on camera. And that brings up yet another little floating palette or little dialog box here. And actually all of those viewing options are visible here. So everything that was under depiction, under viewing controls, side view, camera, effects, lighting, and shininess, all of that's actually available here under this little viewing dialog box. So I might keep this thing open as well. The camera setting lets you switch between a mono camera and a stereo camera. Now we're not going to ever use that. Sometimes you'll actually see like a stereoscopic image where they have two images side by side, and you have to use those special glasses to see like a 3D view of it. I've never come across a reason to use that. But something I do change here, as with a lot of the software that we've used, PyMol has it, Horos has it. You can switch between perspective and orthographic view. The default is perspective. I'm going to switch to orthographic. It probably won't look that much different, but you see there was a slight change in the shape because we've eliminated any sense of perspective. If I had maybe a different molecule that was really long and part of it was disappearing into the distance, changing from perspective to orthographic might be a bigger change. I typically stick with orthographic here in Chimera the same way I do with Pymol and Horos usually. Don't worry about side view now. We'll come back to that later. Rotation, don't worry about that. But the effects and lighting submenus or little tabs here in this little dialog box, these can be very useful. So lighting lets you change your light source. It's much easier to edit your light source here in Chimera than it is in Pymol. First of all, you can switch between light intensity and shininess. So the light intensity just determines, how bright is your light source, and just as importantly, where is your light source? Is it upper left-hand lighting? Is it bottom right? Is it straight on coming from the front? Shininess will control sort of the surface texture of your object. We'll come back to that in a minute. Let's just look at intensity here for a minute. You actually have several choices under intensity. You can choose ambient lighting, where the whole molecule is kind of uniformly lit from all sides. So here's ambient lighting. There's no shadows, there's no depth, because everything is kind of uniformly illuminated. You can choose single point lighting, which is like a spotlight, one spotlight shining on your molecule. And if you look over on the right, you see that little sphere with this little green thumbtack on it. You can pick up that little thumbtack and just rotate your light source around relative to your three-dimensional scene. So it becomes really easy to reposition your lighting by just moving the little thumbtack. You can also control the brightness of that one spotlight. You can have essentially no brightness. You can have more brightness. You can also mess around with the contrast. You can have very low contrast, very high contrast. Switch to 2-point lighting. Now we've got our same spotlight in the upper left, but it's added a second thumbtack. Now we got this little purple thumbtack. We could rotate that around down towards the bottom right to create like a fill light. That's very common in 3D rendering, to have a main light source in the upper left, but then have a fill light just so that your shadows aren't completely black. And there's something here called the key to fill ratio. A higher key to fill ratio means the spotlight is stronger and the fill light is weaker. So you push this all the way up and it's almost like you don't even have a fill light at all. You put it way down and then the fill light is just as strong as your main spotlight. So usually this would be somewhere in the middle, so that our spotlight is stronger, our fill light not quite as strong. There's even a 3-point lighting setup that I don't usually use. That adds yet another thumbtack in here that you can rotate around, but I don't normally do that. I usually stick with 2-point lighting with a main spotlight in the upper left and another light in the bottom right. So then we switch over to shininess. You change from intensity to shininess, and this is going to essentially change the appearance of the texture, the material that's applied to our molecule. And it's going to determine things like how sharp is the specular highlight. That doesn't really seem to be doing a whole lot, does it? Reflectivity, it's not really doing anything. Maybe that would be more. Maybe that would do more if I had just a single light source. I don't know why it's not doing anything. Really not making much change. So it seems like this intensity is the main thing that's changing our appearance. So those are the lighting controls. That's kind of a quick overview of that, but you can actually render some pretty high quality images directly out of Chimera. So I mean, you can render very large, high resolution publication quality images right out of Chimera. So if you want to try doing that, you can actually just play around a lot with the lighting to really exercise good control over that. The final little tab I want to show you here under the viewing controls is the effects menu. There's depth cueing. You might remember that Pymol had something called, I think it was also called depth cueing, but they also called it fog, that made things sort of disappear as they trailed off into the distance. I don't normally play around with that very much. I just leave it at the defaults, but I will click this button to add shadows. Notice that it added pretty significant shadows here. They're actually a little too dark. You can change the quality of the shadows, although it's not really doing a whole lot. I think we might have to go back into the lighting here to change our key to fill ratio. So those shadows look a little bit more realistic. So sometimes you have to kind of go back and forth between lighting and effects to get what you want. But I can turn those shadows off or turn them back on. I don't normally worry too much about transparency or graphics quality, but something else that can be very helpful are silhouettes. What silhouettes do is they add a little black outline. I don't know if you can tell, there's a little black outline around the edges, some of the edges in this molecule. I wonder, some of this might show up better if I change my background color to white. I don't know if that helps at all, but so with the silhouettes, I can add a silhouette and I can change the thickness. And I don't know if the thickness is, I don't know if that's supposed to be in pixels or what the measurements are here. I just know obviously a bigger number is going to give you, a thicker line, but like I say, it doesn't seem to be a straightforward thing like X number of pixels or whatever. So I'm not exactly sure what units the width is using. But those silhouettes, like I say, they add a little bit of a dark outline around parts of the molecule, which can actually be kind of a useful effect. You can have that even if we hide the surface and we have things like ribbons. You can see the little dark outline that we're getting because of the silhouettes on our surface model. You can also see some of the depth cueing, how some of the alpha helices way in the back are a lot paler because of that sense of atmospheric perspective. You can see the cast shadows as I rotate the alpha helices around, they're casting shadows on their neighbors. You might decide that you don't want that in a ribbon model because it just makes it too complicated. If we go back here to shininess, I think maybe okay, yeah, so the shininess control seems to do more when I've got ribbons. It didn't do very much before with my surface model, but you can see I can control a lot I can create almost a kind of a schematic cartoon look if I set, the shininess way down to 0 and the reflectivity, set that up a little higher. I know I can also create kind of a David Goodsell look. I know a lot of people, they just love the look of his work so much that they always try to emulate his work. Let me put the surface back on. Whoops. Let me change the color of that surface to something a little more interesting. So going back under the lighting tab, going back to intensity, I know if we change this to ambient lighting and you play around with the silhouettes, yeah, that's not that great. Maybe maybe if we get rid of the surface and change to a space filling representation. Oops. Yeah, a little bit of that kind of Goodsell looking thing going on. Might be able to change that instead of intensity, instead of setting it here like that, go back to intensity. You know, that's got a little bit of that sort of Goodsell look to it. Could color all the atoms and bonds uniformly. Now that's starting to look like his stuff a lot more. So you can really play around with color, lighting, shininess, all these different effects. To get all kinds of different lighting effects. All right, I'm going to close this one. I'm going to close this protein. Remember, always use closed session. I'm going to open up a nucleic acid real quick. I've got some DNA in here. So with DNA, when you open up DNA, by default, it gives you this thing that looks kind of like a ladder. And we can control that DNA ladder. There's another tool under, where is it? Depiction, right? Nucleotides, because DNA and RNA are made out of nucleotides. They call this particular tool, the nucleotide tool, excuse me. So right now it's showing the backbone of the DNA double helix as this kind of ribbon, but we could change that to atoms and bonds and apply that. So now we've actually got atoms and bonds in our backbone. The sugar and the base. So remember that you've got a nucleotide is made up of a base, like, you know, either a purine or a pyridine attached to deoxyribose, which is a sugar, and then that's attached to the backbone. We can show the base as and sugar as atoms and bonds. So now we've actually changed this to, you know, looking at all of the individual atoms instead of that sort of ladder depiction that we had before. I think there's some hydrogens that are missing, so we can go back to tools, structure editing, and add H, and add some of our missing hydrogens. Didn't like it, added some of them, but keeps giving me these weird error messages. But you know, now that we're looking at this as there's some weird stuff going on here with some of these bonds. Not sure what that's all about. We can change this to, you know, ball and stick or sphere representation. You can see it's still got the black outlines because over here under effects, I've still got silhouettes on, but I can turn those off. I actually kind of like them myself. Again, I can play with the lighting settings, you know, to edit the lighting on this molecule. So there's different things you can do with specifically with nucleic acids, just using that nucleotides depiction tool. One last thing I'll show you is that next week I'll be showing you some of the databases where you can retrieve molecular model files, you know, such as the Protein Data Bank and the orientation of proteins and membranes database, that the OPM database. Within Chimera, you can fetch a molecule directly from one of those databases without even going to the database and downloading it. But you have to know the ID number. So every one of these databases assigns an ID number to each molecule, that's been deposited into the database. And so if you know the right ID number, you can fetch it directly using this option to fetch by ID. And these are all databases over here on the left. We've got the NDB, which is the nucleic acid database, the PDB, which is the protein data bank. There's actually 3 different options here. I'll explain those another time. UniProt is a universal protein sequence database. ViperDB is the virus particle explorer. You can actually download the entire nucleocapsid of a virus, or you can download the one single protein from the nucleocapsid of a virus. For now, I'm just going to show you PDB. And there's a few that I've worked with so frequently that I just know them by heart. There's one called OneTub, which is tubulin. So that's the 4-character code. So if I enter OneTub and click fetch, it automatically opens up a molecule of tubulin. This is a quaternary structure. There are actually 2 different amino acid chains here. A functional molecule of tubulin consists of 2 subunits. There's a molecule of alpha tubulin and a molecule of beta tubulin. And the one on the right here is the alpha tubulin. The one on the left is the beta tubulin. I can tell that because when I hover over part of the molecule, that CYS, it's actually telling me that I'm hovering over a particular amino acid. I'm hovering over cysteine. And that cysteine is number 376. It's the 376th amino acid in the chain. And then the A at the end tells me that it's part of the alpha subunit. And then if I come over here, I'm hovering over a serine residue. It's amino acid number 236 in the chain. And the B is telling me that it's part of the beta chain. So you can get information about the molecule just by hovering over different parts of it. You can tell, which amino acid is at that location and what the number is and which chain you're looking at. So we've got 2 amino acid chains here that are joined together to form one functioning tubulin molecule. So this is an example of quaternary structure. You can see the alpha helices and beta sheets in here. So let's play around with this just a little bit, and then I'll turn you loose to do today's. I still have to show you how to export an image. I'll do that in a minute. Well, let's see. We could go under depiction. Something here, color secondary structure. Remember, that's how we color things by secondary structures. I'll just stick with the defaults and click apply. So now you can see that we've got some red alpha helices, a bunch of purple beta sheets, and we've got the gray, which are just the random loops and coils connecting those things together. Notice there's still some stick models showing. Some of those are other molecules that are embedded within the tubulin, but some of them are also parts of the protein, like some of the side chains of some of the amino acids are visible. I'll show you a different way another time. I'll show you how to hide those. Like, for example, let's say I wanted these other molecules to be visible, but I didn't want the side chains of any of the amino acids visible. If I just go up here to actions, atoms, and bonds and hide all the atoms and bonds, it hides the side chains of the amino acid, but it also hides those other molecules. So I'll come back and show you how to do that another time. And so we've colored by secondary structure. We could add a surface. Yeah, it's giving me another error message, but it seems like it actually did work. It gave me a complete surface. So I could make that surface partially transparent to reveal the ribbons on the inside. I could also color this by chain. Now, if I go to that color all options, I actually don't have a simple option to just color by chain in here. PyMol has that. PyMol has an option to just color by chain. And it just would distinguish the alpha and beta chains of the molecule, but we don't have that here in Chimera. But what we do have is a really robust set of selection tools. And I'll come back and talk about these a lot more later because these selection tools are really powerful. But right at the very top, you can select individual chains. So I could select the alpha chain. And it's hard to tell because I've got the surface and the ribbons in there, but the alpha chain is actually highlighted in green. And so now if I say, okay, let's color our ribbons, let's color them one color. So I just said color ribbons green, but it only applied to the selected chain. So now I go back to my select menu and say select chain. Let's now select the beta chain. In my color thing over here, I can say, all right, let's color the ribbons hot pink in the beta chain. So now I'm able to easily distinguish the alpha and beta chains, whereas my surface is still all one color because in my little color palette over here, I said only color the ribbons. If I wanted to change the color of the surfaces, then I could, change that to something else. Okay, now it only colored the surface on one side because I still have just the beta chain selected. If I go back to my selection menu, I have to say clear selection. Clear selection is how you deselect something that's currently selected. So now if I say color surfaces, it should color the entire surface because I haven't selected just one part or the other. All right, and final thing, just so that you know how to do today's exercise, I'm going to have you open up a small molecule from that sample molecule folder and just do a quick, simple rendering of it. Follow the instructions in the assignment sheet that's posted on my courses. At least I hope it's posted on my courses. I'm not sure I put it in there now that I think about it. It is in there? Okay, good. I think I put it up before break, so then I couldn't remember if I'd actually put it there or not. And then you'll do something similar with protein. Again, follow the instructions. But to actually render an image, you go to the file menu, save image, pretty straightforward. You do have to be aware of a few things here, though. First of all, choose your file type. I'm pretty sure I asked for JPEGs. Yep. So make sure you change the file type to JPEG so you're getting the correct file format. And then I also ask you to make it, roughly 8.5 by 11. Doesn't have to be exact, but pretty close to 8.5 by 11. Right now, it's just giving me pixel dimensions. It's saying, okay, 1590 pixels wide by 931 pixels high. Well, how many inches is that? Who knows? You want to check this little box here that says use print units. Now I can actually put in here my size. Apparently my window is really wide, so it's 11 inches wide. It's not quite, it's not 8.5 tall, but that's close enough. But then I also have the option of entering my resolution. And I think I wanted you to make these 300 pixels per inch. Once again, they incorrectly call it DPI instead of PPI, but we know what it's supposed to mean. So if you check the box that says print units, then you can actually enter, actual size in inches and enter resolution down here. One last thing that I want you to be aware of, you don't necessarily have to do it for today's exercise, but there's a little box here to create a transparent background. That's especially useful if you're saving it as a PNG file, because it will literally make the background transparent. But even saving as a JPEG or a TIFF, I believe if we bring that into Photoshop, it'll let us actually select the molecule independently of its background. So that transparent background checkbox can be very important. So that should have put a JPEG here on my desktop, which it did. If I open that up in Photoshop, let's take a quick look at it. So there's the image in Photoshop. I just want to check something. Okay, so it didn't bring in a selection to select transparency. So the thing that I was looking for doesn't seem to work in JPEG format. Let me see what happens if I mean, I want you to save a JPEG. I'm just kind of trying to remind myself here. If I save it as a TIFF and bring the TIFF in here. Okay, yeah, so if I save it as a TIFF, you see that it really does bring it in with a truly transparent background. So then if I have another layer and I put some other object or something in the background, you can see that the image, the TIFF image really is transparent, and I can put things behind it and you can actually see through them. So the transparent background thing doesn't really work with JPEG, but it does work with PNG files or TIFF files. But as I said, you don't have to worry about that for today's exercise. Just export them as JPEGs. So that's everything. Oh, one last quick thing. We'll come back to this again later too, but you can also export Chimera models in 3D. I showed you that PyMol will let you export a VRML or an STL file, which you can then bring into Maya or Cinema 4D. Similar thing here in Chimera, although it's kind of a weird, you wouldn't expect it. It's actually under export scene is how you export a 3D model. And we have several file types. You can actually export directly to an OBJ, which you could bring into Maya, or you can export to an STL or a VRML that you could bring into Maya or Cinema 4D or Rent or Blender or any other 3D program you wanted. One thing to be aware of though is that it gives you little warnings down here, because sometimes when you export something in a certain 3D format, it won't export all of the features that you've selected. So if I choose VRML, it doesn't support, clipping planes and things like that. It doesn't bring in the depth queuing, or if you had created it as a stereo image, but that's okay. I don't care about that. Most importantly, it's going to export the whole model. But I do think there is a problem with OBJs. Yeah, with an OBJ, it's only going to export the surface model. It's not going to export ribbons. It's not going to export individual atoms. So if you were viewing this as a ball and stick model or a CPK model, it's not going to export any of your atoms. It's only going to export surfaces. So, so OBJ is actually a very limited format. So VRML and STL are actually much better choices because they export the whole model, whether it's, atoms, bonds, ribbons, surfaces, everything. All right, so you got about an hour left to just play around with Chimera and export your models. There are 2 different drop boxes up on my courses, one for the small molecule and then one for the protein. Yes. Yep.
So this is in for this project, right? The focus on the amino acid.
Oh, did I see that? Shoot.
Yeah, we got this.
Yeah, that's in the assignment sheet. I guess I got to show you how to do that.
Well, it says for the actual stuff to do that. But I don't know, is it the actual assignment or?
If it's yep, it is. Yeah, that's the one that's up there. So that's what I want you to do. Shoot, forgot about that. I was hoping to not get sidetracked by selections, but I guess I have to show you how to do that. So for the protein, I wanted you to select a specific amino acid and highlight it. And I forgot that I included that requirement in there. Let me show you how to do that. I'm echoing again. When I say select a specific amino acid residue, when you're hovering over a protein molecule, remember I pointed out that it actually tells you what amino acid that is that you're hovering over. In this case, it's hovering over a threonine and it's number 188 in the chain and it's part of the beta chain. You can actually select a specific amino acid by holding down the control key and clicking. And notice what it's done is it's highlighted a little patch of that alpha helix in green. That little highlighted area represents a single amino acid that I've highlighted. And I ask you in the instructions for the second part of the assignment to draw attention to one particular amino acid in here. And there's a couple of ways you could do that. I mean, you could do something as simple as just change the color. Let's pull up all options here. You could change the color of the ribbon just in that one selected area and make it stand out. Or something else you could do is you could actually change the representation of that and you could actually turn on, say, the ball and stick. You gotta show it. Now I'm saying show atoms and bonds, but since I've selected just one amino acid, if I say show, it's only going to show the atoms and bonds that are part of that specific amino acid. So what you're seeing here is the side chain of that threonine that's sticking out at us. And then, I could color the atoms and bonds. I can color them by element, which they already are, or I could, give the atoms and bonds, some weird color just to highlight them. So that's a way to draw attention to a particular amino acid. So just to repeat, you just hold down the control key and click on any part of the chain and you can highlight a specific amino acid. And like I say, you can, color the ribbon a different color. You can show the atoms and bonds, to see the side chain. You can color those atoms, color them by element or give them some bright color so they stand out from the rest of the molecule, whatever you want to do. I wanted you to do this as part of today's exercise because there are definitely going to be times where you might want to do this. You might actually want to highlight a specific amino acid in a protein to call attention to it. So for example, the final project in this class involves illustrating the molecular mechanism of the disease. One of the diseases that you might choose is sickle cell anemia. And sickle cell anemia results from a mutation of a single amino acid in one of the chains of the hemoglobin molecule. So if you look at normal hemoglobin and you compare it to hemoglobin S, which is the sickle cell form, you look at them side by side, there's no way in the world you're going to be able to tell the difference. But if you highlight the fact that it's glycine changes to an asparagine or something, I forget specifically what the change is, but if you highlight the specific amino acid in the normal hemoglobin and the hemoglobin S, the sickle cell variant, then you can actually see and draw attention to that one little acid change. So that's why I want you to learn how to select individual amino acids. Highlight them. But we'll learn more about selections in Chimera later. There's a ton of really powerful stuff you can do with selections. I don't want to take the time to do it today.